So now we move forward to the workshop. You know that the recent discovery of the CRISPR-Cas9 system has allowed to improve incredibly the tools to modify genomes and allows many applications in different fields. However, this technology has been used recently by a Chinese team to modify human embryo with the idea to inactivate a gene for a genetic disease, but still it raises many crucial issues, scientific and ethical issues. So you know that many papers have been published, scientific and non-scientific papers in the recent weeks, and this raised really a strong debate. So we have decided together with the Japanese Society for Gene Therapy and the European Society of Gene Therapy to propose this roundtable with experts of gene therapy and bioethics to talk about this point and have your comments eventually. So I want to announce the people that will participate to that debate, and Seppo will make an introduction, of course, of the field. And people who will participate are Bernard Bakshi from the Institute of Biomedical Ethics from the University of Geneva, Professor Kazuto Kato, who is Professor of Medical Ethics and Public Policy from the University of Osaka, Robin Lovell-Badge, who is head of the Division of Stem Cell Biology and Development Genetics in Crick Institute, Luigi Naldini, who is director of the San Rafael Teleton Institute, and Adrienne Thrasher, who is professor in pediatrics immunology from the UCL Institute. So we will start with a small introduction, and then we will have this roundtable discussion.
Okay, thank you, Natalie. So I have 4 slides that just captures the topic here. So Chinese colleagues did publish this paper that you may realize it was received March 30 and accepted April 1st. So any editor would envy this fastness of review. But anyway, so CRISPR-Cas technique was used on human 3' pronuclear zygotes. And what did they do? Just briefly. So they wanted to edit beta-globin gene, obviously trying to treat thalassemias or see whether this technique would be capable of doing that. And they took 86 non-viable human embryos from in vitro fertilization treatments and used those for the studies. So these were something that were not used for the IVF therapy. Out of those 86, in 26 cases, they were able to show that the target gene was cleaved successfully. But in these cases, still the efficiency was only around 50%. And they then showed by high-throughput sequencing, whole genome sequencing, that there were substantial off-target effects and mosaicism in these treated embryos. In the paper, they don't discuss ethical issues, but they do mention in the methods the following. This is directly from the paper. So there is a title, Compliance with Ethics Guidelines, and all the authors declare that they have no conflict of interest. And then they say that the study conforms to ethical standards of Helsinki Declaration and national legislation, and was approved by the Medical Ethical Committee of the first affiliated hospital, San Yat-sen University. And the patients donated their 3' pronuclear zygotes for research and signed informed consent forms. So the ethical issue was touched upon on the paper. The general conclusions by the senior author, Jinju Huang, was that he really has concluded that this work is too immature to pursue clinically. That's clear because efficiency was low and a lot of target effects were found. The concern, of course, of most parties is the implication of the germline as an object of genetic alteration, pushing the limits of human genetic modification as it currently stands. Gene editing of human germline has broad scientific, social, and ethical concerns, and that's why we called upon this discussion and debate here. And of course, everybody can imagine questions. Where do we stand in terms of regulation? What should be done or not be done? What can be accomplished? What are the social ethical implications, and how should we move forward? So with that, then Natalie and myself, we want to call upon the panelists to come up onto the podium.
Professor Robin Ali is going to moderate this debate. Thank you, Robin.
So, thank you to the panelists, and I'm going to be moderating this debate. So, and try to. It is a debate, so trying to stimulate some discussion, and I'm going to call upon the panelists to open with a statement, no longer than 5 minutes. And then after the panelists have all opened, we can then start the debate. So I'd like to call upon Kazuto Kato to start with his statement.
Is this on? Yep. Thank you. Well, it's my great pleasure and honor to participate in this very important session. Please allow me to read my memo to keep my speech short. So I'd like to begin by reporting 3 things from Japan. First of all, well, as has been asked in the slide, Japan, well, talking about the guideline, Japan has governmental guidelines for the clinical research of gene therapy. And they clearly, well, a set of guidelines, they clearly prohibit clinical research that aims at modification of human germline genome. However, it does not have a clear guidance about basic research using human eggs and germ cells and so on. Secondly, as many of you, I hope, already know, the joint statement on human genome editing was published in August by American Society of Gene and Cell Therapy and Japanese Society. The statement supports the strong stance against gene editing and modification in human cells, which could lead to heritable germline genome modifications. In particular, the statement points out that conducting clinical research in an ethically accepted way is not possible because effect of modification will have to be analyzed in many generations to come. And the Japanese society played a very important role, and the chairperson of the society, Professor Yasufumi Kaneda, who is in this room, had organized a press conference, and it was broadcast nationwide in the news of NHK public TV program. And there were some other newspaper articles about genome editing. And before the publication of the statement, the Japanese society also held a public forum focusing genome editing in Osaka on the 26th of July. The forum may have been the first of this kind in Asia, and it was my pleasure to be involved with both of these occasions. The third thing to report is that our governmental committee the expert panel on bioethics of the Council for Science, Technology and Innovation Policy in the Cabinet Office of Japan began investigating this issue and held hearings from 3 experts in July and September. I'm one of the members of the panel, and I think we will discuss if we can issue some document in the next meetings in October and later this year. So that's a report about the Japanese situation. Well, how do I think about these issues myself? Well, my overall stance is similar to the joint statement of the 2 societies, American and Japanese. I am against germline genome modification and also support the stance to concentrate on animal experiments first before conducting experiments on human embryos and germ cells, because the technology is so immature. In addition, I think we have to consider several other things as well. I'll quickly tell you 3 things. I believe that it is quite important for scientists and medical researchers, I mean those who are in this room, to discuss and report to the society how much you can do by using genome editing technology on somatic cells without going into controversial area of germline genome modification. We, or you, may want to recall a huge debate we had about the use of cloned human embryos for making ES cells, so-called therapeutic cloning. However, after the appearance of iPS cells, no one is talking about therapeutic cloning because it is no longer required. So if we can do everything, many things with somatic genome editing, that may be good. Secondly, some people argue that inability to obtain consent from future generations have existed in other medical technologies, such as reproductive technology. I argue that this time we are talking about modification of the content of the cells, things within the cells. That is genome, a huge macromolecule. Therefore, we need extra caution before making decision to go ahead. And lastly, it is very important to recognize that if one nation allows, or even one group of researchers conduct heritable human genome modification, effect will spread to the world. So it is different from other forms of medical applications where we seek consent from and analyze results of individuals. So I told you my stance, and I told you what to consider when we have wider social discussions. Thank you for your attention.
Thank you. Thank you. So now I'd like to call on Robin Lover-Badge to give his opening statement.
Okay, well, thank you very much for the opportunity to contribute to this very important debate. So first of all, I should say that I disagree with proposals for a moratorium, in which any case I think would not be effective. With respect to using the methods for research on early human embryos in culture, I would argue that this should not only be permitted, but that it should in fact be encouraged in some circumstances. Much of our knowledge of early development of mammals comes from studies of mouse embryos, yet it is becoming increasingly clear that gene activity and even cell types are very different in human embryos. Genome editing techniques could be used to ask how cell types are specified in the early embryo and the nature and importance of genes that are involved in that process, those processes. So understanding gained could lead to improvements in embryo development following IVF, to reduce implantation failure, and to reduce the incidence of miscarriage. So, and this could rely on techniques that do not themselves involve genome editing, so more conventional treatments. In the context of research on embryos in culture, genome editing techniques are just another method among many that are already in use to help us understand how human development begins. The only thing that makes them special is their likely precision and efficiency. This is justification in itself for why they should be used. Similar arguments can be made for the use of the techniques to study the development of human germ cells in vitro. We know through the work, for example, from Azim Sarani, that there are genes involved in the early development of human germ cells, such as SOX17, which are not required in the mouse. So that's a very clear difference in terms of the genes that are involved. It would be possible to do work on, for example, on spermatogonial stem cells in culture and explore the role of genes and, in fact, epigenetic changes to male infertility, which is increasing in many countries. As to germline gene editing being used to allow children to be born with heritable genetic alterations, I also think it is permissible to carry out relevant research to explore if this might be done safely, but in my view, it is far too early to contemplate doing this for real. There needs to be a frank and open discussion about the many potential uses, which could be correcting genetic defects, mild forms of enhancement, such as providing disease resistance or reducing disease risk, through other types of enhancement that copy traits that are already found in humans, to the extremes of introducing new traits from animals, plants, or synthetic genes. In each specific case, there needs to be detailed information about risk, and there would need to be societal backing before any application should be permitted. However, the mere prospect that genome editing may one day be used to create genetically modified humans should not in itself cause panic or disgust, especially if the aim is to cure or prevent serious disease. In the words of Sarah Chan, who's a colleague on the Hingston group, where we debated these issues a couple of weeks ago, restricting research because of concerns about reproductive application is premature and dangerous, and will ensure that it remains forever premature and dangerous, for want of better knowledge. So it is, in my view, important that this research progresses in places and by people who respect the value of excellent, well-conducted and properly scrutinized research, than see the proliferation of, for example, rogue clinics offering dubious or dangerous services, as has blighted the stem cell field in many ways. I've heard comments from people in Europe, such as, why should we bother debating this topic? It is already illegal in Europe, except perhaps for countries like the UK, which I can come on to in a second. I don't think this is a sensible approach. It's a much broader international issue that needs an open debate, one that engages the public as much as possible. We risk losing perfectly respectable applications, but the dangerous, trivial, or ego-driven applications will probably go on if there was a moratorium, regardless, in jurisdictions with no rules. I should just say, in terms of relating what goes on in the UK, you can do research with a license from the HFEA in the UK on early human embryos and germ cells. You would not be permitted to transfer any embryo into a woman. And in fact, several of the research funders in the UK, notably the Wellcome Trust, MRC, and BBSRC, and the Associated Medical Research Charities and the Academy of Medical Sciences, put out a statement about 3 weeks ago saying that they're open to consider research in this area. So in the UK, at least, we hope there will be properly conducted and research and done in an open and well-scrutinized way. Thank you.
Thank you very much. So I now call on Bakshi for his statement. Thank you.
Thank you very much for the invitation. as I am a philosopher, I will be more general and try to to give you a picture of the ethical landscape. In 2004, the Royal Society published a report on nanotechnologies where ethical questions were divided in 3 categories. First, human dignity concerns. Second, safety and security concerns. Third, social justice concerns, including techno divide, a question of justice, rich countries, poor countries, and so on, and implication for civil liberties, and finally, human enhancement. This scheme is, in my opinion, useful for all technologies, so for gene editing too. As we can immediately see, some concerns grow together. For instance, answers to security concerns tend to put civil liberties in jeopardy, and others not. Since CRISPR is cheap and easy to use, it is not limited to rich countries and will diminish the techno divide. In the debate on germline gene editing, safety and security concerns are in the forefront, especially when we consider gene drive. A problem with these concerns is that they are ethical only at the margin. Safety and security should be guaranteed or provided, but how to do it is a technical and an empirical question, and the debate regards mainly the putative risks and measures to prevent it. 2 normative points are nevertheless implicated in the risk problem. First, should we balance risks and benefits, or should we absolutely avoid certain risks, even if the price is to forego huge benefits? The first stance was adopted with the nuclear industry, but it is changing now, and for some, gene technology is a risk that should not be run at no price. I don't think it is a wise option, but risk aversion is a landmark feature of human psychology. First, a second point, if this last option is nevertheless chosen, as it is the case in our societies, and a threshold of acceptable risk is the solution, who will set it and how? Experts or through a democratic procedure? In safety concerns, we can include the environmental ones. If we use gene editing coupled with gene drive to modify animals and plants, and microbe and virus and so on. This is the first cluster of problem, security and safety. The second is human dignity concerns, and sometimes human enhancement are often linked. It is not new. The old debates concerning embryos' moral status and liberal eugenics, germline modification, were already for a part about these concerns. They re-emerge now and will not find any definitive solution, since they manifest 2 opposite conceptions of human being and of human action. In using gene editing, are we exercising our legitimate powers? Human beings are endowed with rational faculties that prompt them to modify their environment and themselves? Or are we playing God? Maybe our power is legitimate only when we use it with a therapeutic goal, but not for enhancement. But how to justify this claim, and where is the dividing line? Where does gene therapy become genetic enhancement? These questions are well known, as is well known our inability to answer them or to agree on the answer offered. This philosophical disagreement will grow, since gene editing will be used worldwide in cultures very different of ours. Think of China. Consequently, a solution should be global and political, not strictly ethical. Till now, I have spoken of gene editing in general, that aims to modify human and non-human genes. If now I restrict its scope to human genes, a distinction should be done between somatic and germline modifications. With germline gene editing, the risks grow importantly. For example, Jennifer Doudna has pinpointed 5 risks like mosaicism, late onset conditions, and so on. You know that much better than me. If some error happens with germline modification, it will be passed on descendants, a major risk for them and maybe for human species if gene drive is used. There is, of course, a serious ethical problem to run such a risk, especially if comparable benefits are not certain. But there is another ethical problem, akin to the charge of playing God, what the German philosopher Jürgen Habermas has named liberal eugenism. The term eugenism is derogatory and tends to stop moral reflection, far from opening a debate. But the debate is worthwhile because it is not plain that we should refrain from improving our human condition if we could do this safely. In the past, vaccination has aimed at this goal, and with success. Why not use germline modification with the same goal? For example, to delay aging or to soften our aggressiveness, a way to improve human morality. It is a topic that Sarah Chan and John Harris has pondered on, and with this question, I finish my talk. Thank you.
Thank you very much. Now I call on Luigi Naldini to give his statement.
Well, I'd like to start asking whether can we actually do germline editing in humans? Well, not in my country to start with, and probably most of Europe. I mean, making or destroying embryos for research purposes or for testing new technology would not be lawful. Can we do it from the technical standpoint? Probably yes. I would say there's good chance that we can destroy genes or in fact a mutation by the technology that are available. There's much less chances that we could edit a sequence in a safe and effective manner, but we know we have a strategy for doing that, and we are, many of us are working to exploit this strategy in somatic cell gene therapy, and we would like to continue doing this in the best and most profitable manner. I don't think the current state of the technology allows to easily or feasibly transfer to embryo editing, germline editing, or germline editing, whether it's embryo or is a gamete or a germ cell. I think it's far from that. This is not today possible. If you try to put it in practice, you will need selection of those few edited cells. Selecting those cells would require probably add the genetic modification to mark them, which would go really into substantial modification. But this will probably not limit us for a long time. It's quite reasonable that good science will bring forward better strategy and way to possibly enable that. So we have to set, of course, a point where a decision-making strategy, which has to do, of course, with the goal and whether it's ethically or not. And I find hard to find a fully justifiable goal or really moving into this research now. I don't see obvious advantages from embryo or germline editing from a medical standpoint as compared to current strategies of selection or, in fact, at the level of in vitro fertilization. And I think, in fact, most of the strategy that would open up when we enter into this germline editing would really go into those dark areas we've been discussing, augmentation, where we try to imagine that changing a gene would make better your progeny, which I think is far from any reasonable or knowledge of today. So I do think that the key for science today is to continue developing these technologies, which are extremely powerful and promising in the whole area of somatic gene therapy. And this effort, which is fully lawful and reasonable and fully justified, will also identify a potential way to go into future germline correction of genes when that will be feasible and safe enough to be considered.
And finally, I call on Adrienne Thrasher for his statement.
So I wrote a paper in 2003 with Sir Robert Edwards, who you'll remember won the Nobel Prize for development of IVF. And the reason we got together to write that paper was because he heard me give a talk about somatic gene therapy for immunodeficiency. And he felt at that time that these are his words, the optimal time to modify genes for therapy of devastating human disease was either immediately before or after fertilization. Now, so the title of that paper was Averting Abnormal Inheritance, Potential of Gene Therapy and Pre-Implantation Diagnosis. Now, actually, the discussion at that time was relatively simple because we definitely didn't have a technology that would enable precise engineering of the human genome. And the risks of random mutagenesis using conventional integrating vectors was obviously totally unacceptable. But, you know, and so I disagreed with him on that. But, you know, coming at this from the point of view of a physician, which primarily I am, I do see the potential for precise treatment of devastating human disease as long as the technology is mature enough and precise enough, and we can be entirely confident about that. So, to my mind, I do support the I guess I sit a little bit between Robin and Luigi here. I support the development of the technology, absolutely, in all settings, in somatic settings, but I also see the importance of studying gene editing in germline and early human embryos for the reasons that Robin outlined. Now, of course, it has to be very carefully regulated, as it is in the UK. In the UK, we can study human embryos that are not for reimplantation into humans up to 14 days after fertilization. I think society has to decide eventually and across the globe, as other speakers have mentioned, but they can only do that if the science behind all this is rigorous and the public are informed, not only by the scientists but ethicists, as we have represented on this panel. I just want to say just add a few comments about, because there are a lot of current debates and statements in this area at the moment. In the UK, several societies have, such as the Wellcome Trust, MRC, BBSRC, have recently issued a statement. The Hingston Group, which is an international group actually chaired by Robin, has issued a very informative and considered statement, which I think is very well worth reading. And in the US, the National Academy of Sciences is hosting a summit in early December in Washington to discuss these issues. So I'll finish there. Thank you.
Thank you. So I'd now like to open the debate up to people in the audience for comments or questions to the panel. And whilst people perhaps go to we have the first comment or question up here.
So, I mean, this is a very rare thing that it's really needed, because if you are okay with IVF, it's only the case if you have two becoming parents that both have a recessive disease, that you need to do something about it. Otherwise, you can select. Or if you have someone with a dominant disease on two alleles, then you have to do something. So it must be a very rare thing that it's needed.
So, Adrienne, would you like to perhaps respond to that and also add, if I could push you a little bit, to say, under what circumstances could you envisage that if this technology, if it's shown to be effective and safe, might be used, not in the immediate future, but many years in the future.
Well, just to answer your last point, I think you just need to pick out a devastating human disease such as Huntington's disease, where there is potential to eradicate that mutation from the family. The first part, sorry, was again.
Well, it was a response, but in a way, I think you've responded.
I think that's a fair point. I think we need to look at what we are doing today anyway in terms of embryo selection, pre-implantation genetic diagnosis, where we are selecting for healthy human genomes already. And we can, I think we can apply a lot of the rules and regulations that we're using for that to the germline editing field. I don't think we totally need to reinvent the wheel on this.
Robin, did you want to?
I mean, I think, you know, in terms of using PGD to select embryos, you're absolutely right. So this is where you'd be, you know, there will be hopefully relatively few occasions when you're going to have one parent homozygous for a dominant disease or two parents homozygous for recessive disease. But actually, even when you're doing PGD, you have to have sufficient numbers of embryos to select from. And often, and particularly with certain genetic problems, the number of embryos produced by mother in a cycle is very low. So it gets harder and harder, and certainly if you have two or more disease alleles segregating, then again, you can imagine that just doing selection is going to be impossible. So if you can, if you like, cure all the embryos, isn't that a better option than selecting? And that argument has been used by some ethicists just as a principle. If you can cure an embryo, it's better than destroying embryos. I think, though, we shouldn't just restrict ourselves to thinking about the possibility of using these techniques to avoid having children with serious genetic diseases. There are other potential applications, and in fact, from our discussions within the Hingston Group meeting, we didn't go into this in great detail, certainly not in the statement, but there was a feeling that actually the sort of mild forms of enhancement, like disease resistance, were perhaps much more acceptable, and are things that you could think of in benefiting whole communities, societies, where risks of certain diseases are incredibly high. These could be genetic diseases, these could be viruses. So I know there's a trial going on in the US using somatic therapies for HIV. Why not just consider making people resistant to HIV from the very beginning? So I think this all needs debating, but I think don't just limit your thoughts to what particular genetic disease can we avoid.
So that's good. It's a really good controversial point there, Robin. So I can just see the other panelists wanted to jump in. So Bernard, I think you wanted it, and then.
Yes, just a precision here. PGD is less risky, of course, than gene editing, but it is not without its ethical problems. For instance, in Switzerland, where we have just voted to authorize PGD, but it's not done. And in other countries, because you destroy embryos, it is considered as a very huge ethical problem. And I remember the Catholic Church, Pope John Paul II said in the last century that if we could heal the embryos, it's much better than destroy them. And the Catholic Church is not against germline modification with a therapeutic goal, if it is not too risky, of course.
That's a very interesting perspective. And Luigi, and then...
No, actually, I wanted to follow up on this point of, I mean, we have germline editing, or if you want germline modification as a product of science in agriculture for a long time. I think we should learn from that. I mean, this is far from being accepted, and still a lot of hot debate on those goals that were set initially for bringing that germline editing. So I'm very fearful, on the one hand, of the consequence of science moving too fast and possibly triggering a backlash from society and stopping what could be very relevant, actually, progress in the area of somatic, when it's too early to decide. And second, as I said, I do think that any attempt of editing in terms, if it's not augmentation, but if I can take your example, Robin, in terms of this, making an individual HIV resistance, well, I wouldn't want to get rid of my CCR5 receptor throughout all the cell. You know, there's probably, I wouldn't know how to go for. This is really playing God to me, to a certain extent. I think it's far from where we understand the genome function and the interplay with them to bring that on the table today. I think we have a lot to learn about the editing in a somatic cell before we go.
So,
I've just added
So,
one other point. Sorry.
Yeah, can I?
Just quickly, that
Yeah.
if we, just to spin that round, if you think about sickle cell, then sickle cell trait, of course, offers protection, at least some protection against malaria. So you wouldn't want to delete that necessarily from the population.
yeah.
I don't disagree at all, but we just, we're going to get more and more knowledge of what genes do from over the next few years.
Yeah, I first agree with the discussion comments about the importance of diversity of genes. I have a question to Robin. UK seems to be quite aggressive, but I told the audience that the effect will spread all around the globe, to the human population. So when we have diverse society, culture, and opinions in many different countries and societies, where we might have quite slow thinking. Well, sorry, I can't. Yeah. How do you justify your decision to go ahead?
Well, the decision isn't to go ahead and do germline changes. The decision is to allow research in this area, which is not the same thing.
Yeah, I know, but
we have a very clear, in the UK, we have this very good regulations which make it very clear what you can and can't do. And it's scrutinized the whole time. If you break the law, you end up in jail. It's not, so we have good regulations in the UK. What I would like to encourage is other countries, other jurisdictions to have good regulations, and certainly there are many countries that don't have regulations covering this at all. And I think there's nothing to stop this work going on in those other countries. So the bigger and the better and the wider the debate and the engagement of the public in all these issues, I think you're more likely to get sensible decisions made across the world than if you say, we're not going to do it. I think that's not going to help. If you close the door now, close the door to debate even, it's really not going to help. You've got to have it open. And the more you involve the public, and so they understand the risks, they're not going to go off to some strange clinic in a country which has no regulations and agree to do something which is stupid just for reasons of ego. I want to have a child that looks like this. So I think we need to. I would like to encourage regulators to get their acts together. The scientists, I think we know what we're doing. I think it's making sure that the more unscrupulous people are controlled.
So I'm conscious of time, so I think we've had a very good discussion, but I do want to wrap up with, because this is a debate, so I want to put two propositions forward, and I'd like, if I could, from the panel, a yes or a no to each of these two propositions, and then I'm going to turn to the audience and get a straw poll to see where we are in the room. And I think, so the first question really is, do the panel envisage, with the proviso that it's proved safe and effective technology in the future, can the panel conceive of clinical application of germline editing under any circumstance? So without the nuances of whether it's Huntington's or enhancement, but just under any circumstance. So I'd like to go in order and start with
Kazuto, yes. Clinical application. No.
Robin?
Yes.
Bernard?
Yes, but only if the benefits
buts.
are
Yes or
bigger
no?
than the risks. Yes.
Yes. Luigi?
I cannot answer yes or not. I have to explain. So I would go.
No, the question was very simple. The technology is perfected, and it's you can contemplate any clinical application. It may be restricted to one application, or would you rule it out completely, ever?
Yeah, I can see at least one, yes.
Yes. Adrienne?
Yes.
Okay. So so you heard our panelists on the first question. The second question is, should there be, from now, a moratorium on all research involving germline editing on human embryonic stem cells? Again, starting from you.
Well, I think I
Yes or no?
Yes.
No.
No, but moratorium.
No.
Oh my.
No. Okay.
Just one word. Moratorium is on application, not on research.
Moratorium on research?
On research, no, of course.
No. Okay. Luigi?
Yes, of course.
No. Okay. So we've heard the panel, so we got clear yes, no. And now I'd like to ask the audience. So we're voting again. First of all, whether you can contemplate ever, and again, once the technology has been established to be safe and effective, under any, even if it's just one application you can conceive of, should there ever be clinical application? So first, and that's yes. Raise your hands. And then no. So we've got a clear majority for yes. And then the second proposal, should there be a moratorium on all research? If there should be, raise your hands now. Okay, we have a few individuals. And there should be no moratorium? Pretty clear.
But you said moratorium on research on germline editing of the human. Right?
Yes, correct. Yeah. So basically, I think that was a very good discussion. I'd like to thank all the panelists and thank you for participating. Thank you.
Okay, please don't go yet, because now it's time to Helsinki City Deputy Mayor Pekka Sauri to welcome delegates and to Helsinki and to this meeting. Pekka, please.
Ladies and gentlemen, good evening. Don't worry, I've cut my normal 90-minute lecture into 85 minutes. Another straw poll. How many of you are in Helsinki first time? That looks like a majority. Helsinki has been featuring pretty high on various international city rankings in the past 5-10 years, especially vis-à-vis quality of life in a city. This is nice in our neck of the woods. And I think if there's one thing that is behind those quality of life rankings, I think it's a fairly good fusion between reliability and creativity. Reliability is boring, it's predictable, it means that services work, trams come on time, lights come on when you switch them, and so on. Easy. Creativity is sexy, unpredictable, interesting. That's also pretty easy. But combining the two in the right measures, I think that's the Helsinki trick, if there is one. And I hope you'll be able to glimpse some of that idea during your stay here. The Finns, which I think many of you will be encountering the first time in your lives, judging by the straw poll, Finns are pretty well acknowledged for their self-irony, sometimes unintentional, and dry humor otherwise, and I'm sure you bump into that as well during your stay here. It's not your lucky day tomorrow because we're going to have a day of industrial action, not directed at the city, but more to the national government, which means that there will be no public transport tomorrow at all. So I hope your hotels are pretty close to where you're going to have your conference. They also promised some rain tomorrow, so it makes for a perfect day, doesn't it? So anyway, have a happy stay in Helsinki in the Finlandia Hall. And I'm sure you'll find some nice pubs, bistros, and other establishments to continue your seminal discussions on the ethics of gene research. Welcome to Helsinki.
Okay, thank you, Pekka. And now it's time for welcome reception and poster session number 1. So that will all be in Piazza, so when you go out on your right hand. So thank you, everybody. Session is closed.